All right, hello everyone. All right, so you're interested in studying a specific protein. You've already took the time, isolated the protein. You've purified it as well, and now you're ready to really study or do some structural analysis of the protein and run some in vitro biochemical assay. But you're really not sure of the actual sequence of the protein, and you want to make sure it is the actual protein that you're interested in. So, and then comes the idea of protein sequencing. And what it is basically being able to identify the exact sequence of a protein of interest, right? So there's a number of different methods that can be utilized to identify the sequence of the protein. If you're dealing with a protein that may be 100 amino acids long or even longer, it would be a bit difficult to sequence it all as one entire polypeptide chain. So what we do in protein sequencing is we actually cleave a given polypeptide chain into different fragments, right? So one of the first step really is to cleave the polypeptide, right? And what generates, polypeptide, and what we generate by cleaving it is really different fragments, right? We end up with fragments, right? And what fragments are, we're basically referring to the protein that has been cleaved at different peptide bonds to generate smaller peptide sequences or polypeptide sequences. So the very first step is to really fragment it. And there are a number of different methods that can be used to fragment the peptide. Some are enzymes, and some use chemical methods, right? We could fragment it using an enzymatic method, right? We could use an enzymatic or a chemical method, right? So when we use an enzyme, we call those specific enzymes that can cleave a peptide sequence as peptidases or proteases, right? So enzymes that can cleave a peptide bond are really known as peptidases. We haven't yet talked about naming proteins and enzyme, but if the name ends in ASE, we're really referring to an enzyme that can catalyze a specific reaction, right? So peptidases. And just like the name may imply, peptidases, they cleave a peptide bond. There are two different types of peptidases, one known as endopeptidases and another known as exopeptidases, right? Now, what's the difference between endo and exopeptidases? What exactly is happening when we use an endopeptidase or an exopeptidase? So let's take a look at an example of a peptide bond, right? So I am just going to draw the NH, and it's connected to the rest of the protein. And here's the side chain of one amino acid, right? And then we have the carbonyl oxygen. And I'll just draw the next peptide bond, right? So that's the bond that's really connecting the next amino acid or the adjacent amino acid. And here's its NH, it's alpha carbon, and it's carbonyl oxygen. And again, just carries on to the actual peptide sequence, right? So suppose that R is really an amino acid within the sequence, but to identify which one it is, I'm just going to call it Rn, right? It's the amino acid number N within the sequence. It could be like aspartic acid. And right here would be R is N plus 1, right? Whatever this is, this would be N plus 1. All right, well those enzymes do, or what fragmenting a polypeptide chain is involving is really the cleavage or the hydrolysis of a peptide bond. Remember, to form a peptide between two given amino acids, it's a condensation reaction that involves the loss of a water molecule. Well, to cleave a peptide bond, it's a hydrolysis reaction that requires the addition of a water molecule. All right. Well, peptidases are enzymes that cleave a specific peptide bond, right? So, and we call that or refer to that sometimes as the sizzle bond, right? It's the bond that will end up being cleaved. Well, endopeptidases cleave a peptide bond somewhere within the sequence of the polypeptide chain, right? So endopeptidases cleave within the sequence, somewhere, whereas exopeptidases specifically cleave at the N or the C terminus, at the N or C terminus, right, of the peptide, of the polypeptide chain, Meaning if you're using an exopeptidase, it would simply cleave right at the N terminus, that first peptide bond, or right at the C terminus, right, at the end of the peptide bond. And remember, when we draw a sequence, we start with the N terminus end, right? This is really the N terminus end of the peptide, and when we write the sequence, we end with the C terminus end, right? Terminus, right? Where you have the free carboxylate and you have the free amino group. All right, so those are endo and exopeptidases. They're cleaving a specific bond, right? And in a chemical method, one common chemical that we can use is, I'm just going to write cyanogen bromide. And oftentimes we'll see it abbreviated as cyanogen bromide. Okay. So what are some examples of endopeptidases and exopeptidases? We're going to take a look at two examples, right, of really endopeptidases. And again, they're cleaving a specific peptide bond within the sequence, right? And that peptide bond is between N and N plus 1, all right? It's between two given amino acids. Endopeptidases, some tend to be highly specific in that they only recognize specific sequences within the polypeptide chain and cleave at that sequence, right? Some are a little less specific, in meaning that the peptide bond that they're cleaving, those two amino acids can vary quite a bit, right? So in one example of an endopeptidase that we'll encounter is trypsin, right? I'm just going to go ahead and write it here, actually, right? Is trypsin, right? And trypsin is an example of an endopeptidase that cleaves a specific peptide bond within a given polypeptide chain. And it recognizes positively charged side chains where N, right, where N, or Rn, is equal to either arginine or lysine, right? So when the first amino acid within that sequence where the peptide bond is to be cleaved is either arginine or lysine, it would cleave that peptide bond. We say that it cleaves it at the C terminal end of the amino acid, because think about this is the amino group of arginine, or of that R group, and then you have an alpha carbon, and then you have a carbonyl oxygen, and that's the C terminus of that the end of that sequence, right? So it cleaves at the C terminus end of that entire sequence. All right, so once it cleaves it, arginine would end up being at the C terminus, or lysine would end up being at the C terminus of that fragment, right? N+1 could be any amino acid as long as it is not a proline, right? And it has to do with the mechanism of cleaving it. And if you think about proline lacks this hydrogen in its amide bond. That's because the side chain of proline, the nitrogen is part of the ring, right? So it cleaves it as long as the next amino acid is not proline. And we'll actually talk about trypsin quite a bit. And the reason that it recognizes specifically arginine and lysine, or it cleaves after positively charged residue, is the pocket where those particular amino acids will fit in trypsin has a negatively charged aspartic acid residue. So it's like you have the pocket at the active site of trypsin, and that pocket has a negatively charged aspartic acid residue, right? Which will stabilize the positive charge from either arginine or lysine, right? The positive charge of either arginine or lysine at the active site of trypsin. We'll talk about trypsin quite a bit. It also has 3 amino acids that are quite essential in cleaving that particular peptide bond, and we call those catalytic triad, right? There's a serine residue, there's a histidine residue, and there's an aspartic acid residues. They're all present at the active site. And those aspartic acid residues are different aspartic acid residues, right? This specifically has position 102, so it is amino acid number 102. It's like this would be 1, 2, 3, 4, 5. That's 102. The serine, I believe, is 95, and the histidine is 57, right? Notice the amino acids vary apart in sequence, right? One is at 95, one is at amino acid 57, one is 102, but in the folded trypsin, all of those 3 amino acids are next to one another, and they are at the active site of many enzymes that we refer to as serine proteases, right? Enzymes that cleave a peptide bond. And that particular aspartic acid is really at position 186 in the sequence, right? Okay, so this will make a bit more sense once we start talking about the quaternary structure of a protein and how amino acids that vary, that are not necessarily close to one another in sequence are close to one another in a folded protein. All right, so those 3 amino acids are present at the active site of trypsin. And there's this pocket that has a negatively charged aspartic acid residue that will form charge interactions with arginine or lysine, and hence trypsin specifically cleaves positively charged, the peptide bond after positively charged amino acids. So it is highly specific, right? So if we use it to cleave a peptide sequence, we know it will only cleave after arginine and lysine. All right, another example of an endopeptidase is chymotrypsin. I'm just going to use a different color. So chymotrypsin, right? Chymotrypsin cleaves after aromatic amino acid residues, or hydrophobic and aromatic amino acid residues, where Rn is either tryptophan, tyrosine, or phenylalanine. And as long as the next amino acid in the sequence, or as long as Rn+1 again does not equal proline, right? It is a bit less specific than trypsin in that it can also cleave after other amino acids, right? So we'll say that trypsin is, okay, it has a smaller or a lower specificity comparing to trypsin. It's less specific since RN can also be aspartic acid. It could also be a histidine. It could also be methionine, and it could also be a leucine, right? So it's less specific. Mostly after aromatic side chains, tryptophan, tyrosine, phenylalanine, its specificity pocket, we call that the specificity pocket because that's where the specificity of that particular enzyme comes in play, where it only binds or cleaves after arginine or lysine. The specificity pocket of chymotrypsin really is hydrophobic, right? So it would cleave after aspartic acid, tyrosine, or phenylalanine, but again, it's somewhat less specific that it can cleave after aspartic acid, histidine, methionine, or leucine. Okay, well, what about cyanogen bromide? What does it cleave after? It still cleaves a acyl bond as well, but it cleaves where Rn is really methionine. And again, as long as Rn plus 1 is not proline, right? So does every single enzyme or chemical method does not cleave after proline? No, there are some that where proline doesn't really, where this next amino acid can be proline, but those examples, the next amino acid cannot be proline. And it just has to do with the actual mechanism. All right, so I'm going to erase this, and we're going to consider that particular peptide sequence. And we're going to see what happens when we cleave it with trypsin and what happens when we cleave it with cyanogen bromide. So if we have a particular peptide sequence and we're really interested in figuring out the actual sequence of the peptide, what we typically do is we cleave it using two different methods. You could use two different enzymes, right? Or you could use an enzymatic method and a chemical method. So we're going to use trypsin and cyanogen bromide. Why would we use trypsin over chymotrypsin? Because we know trypsin is quite specific. It's highly specific. It's only cleaving after arginine and a lysine. All right, so I'm going to erase some stuff. All right. So, should we erase everything? Erase the ones we don't need. All right. So I'm going to erase the fact that it's highly specific. It does have a catalytic triad, and we kind of already talked about that. But I'm going to go ahead and erase that, right, to make it less crowded. Its specificity pocket, which is, a pocket created when the actual protein is folded, fits arginine or lysine right in the pocket, and they're stabilized by charged interaction with aspartic acid. But again, that's something we talk about in a different chapter. So I'm going to go ahead and erase that. All right, and we're using cyanogen bromide, right? That just said the abbreviation for it. So that's the chemical method that we're using. All right, so now let's take a look at the peptide sequence. How many fragments will it generate? We're cleaving the peptide using a chemical and using an enzymatic method, right? So it's like having two separate reaction mixtures. In one, you'll incubate the peptide sequence with cyanogen bromide. In another reaction, you'll incubate the peptide sequence with trypsin, right? So we are going to call it, really, this is our first reaction mixture, all right? And in our second reaction mixture, we're putting in trypsin, okay? So trypsin cleaves the peptide sequence after an arginine or lysine as long as the next amino acid is not proline. All right, so let's try it. All right, so I'm gonna use the yellow. All right, so every time you see an arginine, and if you recall, arginine is R, lysine is K, all right, and proline is P. So every time you see an arginine or a lysine, that peptide bond is being cleaved right after arginine or lysine, or what we call at the C-terminus end of, at the C end, or the C-terminus end of arginine or lysine. All right, so alanine, lysine, here's lysine, and lysine is not followed by proline. So it would cleave right here, and the very first fragment that we would get in solution would have the sequence AK. All right, and then methionine, arginine, well, it typically cleaves right after arginine as long as the next amino acid is not proline, so it would not generate a fragment right here. And then we have another arginine, and then aspartic acid, but it would cleave right after that particular arginine. It doesn't matter if the amino acid right before it is proline, as long as the one right after arginine is not proline. And again, we're reading it always from the N-terminus to the C-terminus. So now the next fragment would have the sequence methionine, arginine, proline, arginine. All right, methionine, arginine. All right, methionine, arginine, proline, and arginine. All right, that's our second fragment. And then we can keep on going. So we have aspartic acid, we have tyrosine, we have methionine, we have lysine, right? After lysine, the next one is histidine, so it will cleave right here, and then we have aspartic acid, tyrosine, methionine, and lysine, all right? And then we have histidine, leucine, glutamic acid, proline, methionine, alanine, and arginine. So here's an arginine, so that's a longer sequence. It would be H, L, E, P, M, A, R. All right, and then our very last one is glycine, so it would simply be G. I'm actually gonna add an amino acid. I'm going to say GAA, right? So it would be GAA. And now the C-terminus end is really at the end. Okay, GAA, right? So incubating the peptide sequence with trypsin cleaves it into 1, 2, 3, 4, 5 fragments. What do you notice about those 5 fragments, right? Ends in lysine, ends in arginine, ends in lysine, ends in arginine. It'll always end in lysine or arginine except at the C-terminus end of the peptide bond. So unless the C-terminus amino acid is arginine or lysine, it will always be different. So that's sometimes a hint. It's the C-terminus. I'm not sure if you could see this. Right? It's a hint that it's the C-terminus end. Again, because it's not arginine or lysine, and it's always cleaving right after arginine or lysine. Right? So the last amino acid then in the sequence is either arginine or lysine. Had this been an arginine or lysine, then it would have been a bit more difficult to identify the C-terminus end. All right. So now what about if we're using methionine? And I will try and use a different color. All right, to show where it cleaves. So it cleaves after methionine as long as the next amino acid is not proline, and methionine is an M. Okay, so let's take a look. Actually, I think blue will do. So methionine, here's a methionine, so it would cleave right here. This is not proline. And if you take a look, here's a methionine. This is not proline. It would cleave right here. And do we have another methionine? I mean, it's preceded by a proline, but that doesn't affect it. So here is another point of cleavage, and that's it. So it would only cleave it into 3 fragments. And again, one methionine, another methionine, and another methionine, and none of them comes right. You know, right after it is a proline. All right. So the 3 different sequences that we get are, you know, sorry, I keep switching colors, but so we would get A, K, M, R, P, R, N, Y, M, and then we would get K, H, L, E, P, M, and then we would get A, R, G, A, 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 R, G, A, A, right? So if it cleaves 1, 2, 3 times, we ended up with 1, 2, 3, 4 fragments, because the very last one, if it cleaves at 1, 2, 3, 4 sites, 1, 2, 3, 4 sites, then again we get the very last one, so we have a 5th one. All right, and if you notice again, all of them end with methionine, right? Except this one, it doesn't end with a methionine. That's a hint that it's at the N-terminus end of the peptide sequence. So if you're actually given the fact that you're trying to identify an actual protein sequence and it was sequenced using 2 different methods, cyanogen bromide and trypsin, and those are basically the fragments that generates, and you're asked to reconstruct the actual sequence, then those hints would end up being helpful, right? You would know what chemical, you would know if it used a chemical or an enzymatic method based on the very last amino acid. All right, in each fragment. That would give you the specificity of it. If it's methionine and only methionine, you know it's the cyanogen bromide. If it ends with lysine or arginine, then it is trypsin, right? And it depends which other endopeptidases or exopeptidases did you really need to know, all right, or you were asked to know by your professor. Okay, so what if we wanted to construct the sequence? So suppose we don't actually know the sequence, and we were given those and we were asked to reconstruct the actual sequence. Then we would know that we used cyanogen bromide because it ends with methionine. We would know that we used trypsin because it ended with lysine or arginine. And then the hint is really that very last amino acid that was different. So we know that that's the C-terminus end of the peptide sequence. So we can start working backwards. And the way we would do that is you could tell that you have alanine, alanine, glycine. We have alanine, alanine, glycine, but then we have arginine, alanine, right? And if we use trypsin, one of the end 2 amino acids in one of the fragments right here would have to end with arginine and alanine. And if you take a look, here's arginine and alanine, right? So that has to be the next amino acid sequence. So if we're writing it, then the C-terminus end would have been AAGRA, MPELH. But there's a methionine right here. So which fragment of the reaction with cyanogen bromide does it end with MPE? You just find MPE, okay, and then MPELHK, but we have the K, but we can't really know which K it is, right? We have 2 Ks, but we're really not sure which one it is, and you can start working from the front. So we know this is the very last fragment. We have alanine, arginine, and lysine. We have alanine, methionine, aspartic acid, and histidine. The only 2 that actually match are the ones with the alanine. So what does that amino acid have to be at the N-terminus end, right? So it's like a puzzle in a way. Right? But those are not scrambled. They're written in order, so in order of the peptide. But if you're given something like that, they could be, mixed in, so you have to figure it out yourself. That's how we look at it, and then we can reconstruct it and get the original peptide sequence. So are those the only two methods that are used in protein sequencing? No. So one step is to take two enzymes and cleave it, right? So it's going to cleave and generate fragments using trypsin. It's going to cleave and generate fragments using cyanogen bromide. We can align them and determine the sequence. But what if we didn't know the sequence of any of the amino acids in any of the fragments? Then we can separate the fragments and we can identify the actual sequence using Edman degradation, for example, right? So what if you were able to separate those different peptide sequences, fragments using chromatography, you can identify the sequence of each using Edman degradation. And if you guys recall, what we said about Edman degradation is that it cleaves at the C-terminus end of a peptide sequence, and it cleaves one amino acid at a time. So if you have a peptide sequence that has 13 peptide bonds or 14 amino acids long, it would require 13 cycles of Edman degradation. And in each cycle, you can identify the N-terminal amino acid. All right? So what can we say? We're going to just kind of mention Edman degradation. The other one that can identify the N-terminal amino acid residue is using dansyl chloride, which is a fluorescent compound. It specifically interacts with epsilon, or with primary amino group. So it'll interact with the amino group at the N-terminus. If we have a lysine, it will interact with the N-terminus with the epsilon amino group of lysine, and those will basically, those amino acids will fluoresce. We'll have a fluorescent, and we can identify how many we have. We typically use those methods to identify if we have a protein that is composed of more than one polypeptide chain, and we'd like to identify how many polypeptide chain it has. We do the N-terminus analysis. But Edman degradation is great because it only cleaves the very last, it only cleaves one amino acid at the N-terminus. Whereas if you use the dansyl chloride, at the end it's under acidic conditions, which causes all the peptide bonds to be cleaved. But the only ones that will fluoresce is the amino acid at the N-terminus and any lysines. All right, so we're going to take a look a little at Edman degradation. This is not it, and this is not it either, so let's try the blue. Okay, so what you have is a compound, nitrogen, carbon, and a sulfur, all right? And it specifically interacts with the N-terminus end of a peptide sequence. So we have a lone pair. It actually needs the lone pair. So that's just the peptide sequence, and we'll call that R1, the very first amino acid, right? P. It is known as a phenyl. P. I-T, phenyl isothiocyanate. It's called PITC, phenyl isothiocyanate. All right, that's the reagent that is used, and it reacts with the N-terminal of the peptide sequence. And what happens is, under acidic conditions, it forms a PTC, all right, polypeptide adduct, right? So polypeptide. So it basically interacts with the amino groups and is connected to the polypeptide. And then what we use is some acidic conditions, right? And what we get is what we refer to as a PTH connected to the amino acid, and it's specifically only connected to the N-terminal amino acid residue. It will cleave it from the rest of the peptide, but the rest of the peptide will remain intact. And how that PTH amino acid adduct basically, here's the phenyl thiocyanate, right? And I'm just gonna change that peptide into pink for the sake of showing you what happens. All right, so what we get is an adduct where we have the nitrogen, we have that phenyl group, we have the carbon that's connected to the sulfur, right? Not sure that you see that. I don't think you see that. All right, I'll write smaller. So we have the nitrogen connected to the phenyl group, which is connected to the carbon, that's connected to the sulfur. And then we form this bond to the actual peptide, right? This is connected to the nitrogen that was at the N-terminus end, which is basically connected to the alpha carbon. That alpha carbon is connected to the carbonyl oxygen that now forms a bond to that nitrogen, and that's the peptide R1 right here, right? So it forms what we call this PTH amino acid, right? So suppose, and that's one cycle of Edman degradation. So suppose you're running, you're trying to determine the identity of each amino acid in that particular sequence. We'll just take that as an example. I'm actually going to erase all of these because we're done with that particular problem. Right? So suppose you're trying to identify the sequence using Edman degradation of a peptide that has the sequence MRPR, right? After the very first cycle, it's going to form a PTH amino acid adduct, so we would get PTH forming an adduct to methionine, right? And then what else do we get after the first cycle is the rest of it. RPR, right? That's just the first cycle of an Edman degradation. And if we run 2 cycles, right? If you're running another cycle and you're keeping everything in solution, what you now have is, or really at the second cycle, so you ran the first cycle, you basically took out the PTH methionine adduct, and what you have left is here. So then you get a new PTH R adduct and the rest, which is P and R, right? And at the third cycle, you would get PTH proline adduct, and you'd get arginine on its own. So if you have a peptide sequence that has 1, 2, 3 peptide bonds, right, it would require 3 cycles of Edman degradation to completely be able to identify each amino acid in the sequence. All right, well, I hope this was helpful. See you next time.